All right, so in the last part, we kind of went through and talked about some of the enzymes that make up the DNA synthesis machine, and now we're going to kind of get into some of the details of the DNA replication. So, what you have with DNA is you have a leading, or DNA replication, you have a leading strand and you have a lagging strand. And with the leading strand, you only need one primer. It's synthesized continuously, and with a lagging strand, you need multiple primers because what you're doing is you're synthesizing it discontinuously, and you're giving rise to a bunch of what are known as Okazaki fragments. And this is named after the Japanese researcher who kind of discovered this process and laid out the details for it. Okay, and I know, kind of seems a little foreign to you right now in terms of all of these big words being thrown at you and this process, but as we go through it, I'm sure you're going to understand it by the time we're all done. And it's important that you guys ask questions when you have them and make sure that you're writing them down so that we can discuss them in class. Okay? So now we talked about the DNA polymerase and the idea that this is going to catalyze the formation of the two new strands so that we get the DNA copied, and they're going to do so at the replication fork. And it's important again that you keep in mind here that DNA always is growing from the free 3' end. Okay, so it's growing 5' to 3'. So in other words, if you look here, this is the leading strand. Okay, here's the 5' end. It's growing in this direction, and this is the 3' end. Okay, and then this is discontinuous. This is the lagging strand, and we'll talk about this here in a minute. But again, this is the 3' end. So down here, this would be 5'. So we're going to actually grow in this direction right here. Okay, so we're going to kind of concentrate on those. And I know it seems weird, but again, just kind of hang with it, and I think you'll be able to get through it. But one of the things I always get, one of the questions I always get is, why does it only add to the free 3' end? And one of the important things is to remember with this whole process is it requires some energy in order to synthesize the DNA. And the way the cell makes use of energy is by these triphosphates. Okay, so for instance, if we look right here, you can see that there's 3 phosphates. So if we break these things, we're of course going to liberate some energy, and that energy is going to be used to drive the synthesis of this reaction. Okay, so this right here, it's important that you understand this is the hypothetical kind of way. This is not the way that it occurs, but you have to explain why it doesn't occur. So I need you to understand kind of what's going on here. And it's important to note that one of the things we talked about in the last video was DNA can be synthesized in bacteria 1,000 times a second, 1,000 base pairs per second, but it's only 50 base pairs per second in a eukaryotic cell, and that's because the eukaryotes have a proofreading mechanism. So in other words, if the wrong DNA gets, or wrong nucleotide gets added, the eukaryotic cell has a way to kind of correct for that. Okay? So if we look here, you can see if this is the incoming nucleotide, you can see the triphosphate right here. And if we add it on, what we have the ability to do is with this triphosphate right here, we can break this bond and we can add this on. So in other words, this right here is where this bond was broken, liberating these phosphates and adding the new one right there. And then the thing to keep in mind is if that's the wrong one we discover through proofreading, we have to take this one out. We have to get rid of it. Well, if we get rid of it now, we've got no triphosphate there. There's no energy. Even though we have one on this incoming one, there's no phosphate right here. So we don't have any way to add it to it. So DNA replication would stop and we'd have to start all over again. Now, in terms of the actual way it happens, this might help you understand kind of what's going on right here. Okay? So in other words, if we bring this nucleotide in, you can see we've got a triphosphate just like we had over here, but if we bring it in and we join it up and now all of a sudden we find out right here that it's wrong, we have a way to proofread it and get it out. In other words, we take it out and now we're right back here just like we started right up here, and our triphosphate is on the correct side. So now here's our energy source. When we break that bond, we can join it right here and we get rid of those phosphates and the process continues. So in the actual 5' to 3' growth, adding to this free 3' end right here, what you have is the phosphate on the correct side, you have the energy source on the correct side so that you can continually add the nucleotides, and if you make an error, take it out, and you still have the energy on the correct side. So if you have any questions with that, make sure you write them down, and I'll be happy to go over it with you guys more in class. Okay? So now we say that the two strands of DNA are anti-parallel. In other words, they run in opposite directions. This one here, for instance, if you look, you see it goes from 5' to 3'. And this one here, same way, you go from 5' to 3'. Okay, and then a replication fork is going to go in this direction right here. All right, so we can only add them to that free 3' end. So when you're looking at it, again, we have a primer that we would add on right here. Okay, and that's going to get the process moving in this direction. So this is the 3' end, and we can continually add in that direction. Okay, now when you have the lagging strand, here's our 5', here's our 3'. So if this is the 3' on top, what we have over here is the 5'. So we have to add it in this direction. And when we get to here, we got to start another one down here. 5', work in this direction to 3', and we got to just keep adding those on. And what these little fragments are known as is as the Okazaki fragments. Okay, so when we open up this replication fork, that's where DNA polymerase III jumps on. It starts adding the nucleotides in the correct spot according to the base pairing rules to that free 3' end. And what it might look like is something like this if you were to look at it under an electron microscope. And I've got some other videos that I can show you in class too. But for the time being, it's just important to know that all of those different types of things you can kind of look through here and see all of these different types of enzymes that we were talking about right through here. All of these different things are making up this DNA replication machine. Okay, so it's important to know something about the accuracy or the fidelity of this. All right, so if you guys just think about something for a second, if you were able to type 8-letter words, 60 words per minute, how long do you think it would take you to match the accuracy of your cells in replicating the DNA? Okay, so just think about it. You can come up with some sort of an answer. Again, 8-letter words, 60 words a minute. Okay, how long would it take you to match the fidelity, the accuracy of the DNA replication inside your body? Okay, so if you come up with some sort of an error, or some sort of an answer, you'd be allowed to make an error once every 39 years. Yes, 39 years. So it seems like, what? So this is very, very accurate because what you have is the proofreading mechanism that we've been talking about. These initial errors occur in a rate of about 1 in 100,000, but you get errors about 1 in 10 billion. Okay? So the reason errors occur in our body is because we're copying, every time we replicate our DNA in every cell, we're copying 3 billion base pairs. So, errors do happen, but they don't happen very often. So it's very important to know that this is a very accurate process that occurs inside your cells, okay? And what the proofreading does is basically checking the DNA to make sure that the base pairs are matching the way they do. And there's enzymes that go through and do this. And, of course, it's not 100% effective. Sometimes you get these errors that have been perpetuated and it might end up as cancer or something like that. But by and large, your body does a pretty good job of this. Okay, you might have some environmental types of radiation or pollution or x-rays or something like that kind of creates an error in the DNA, and your DNA polymerase and your DNA ligase have the ability to go through and fix these problems and do so effectively so that you don't get cancer or some other sort of a mutation. Okay? So that pretty much, sums up the DNA replication. And what we're going to do is go through and talk about this a little bit more in class. So if you have any questions, make sure that you ask them, write them down, and then we'll watch a couple of these videos. Okay? So we'll see you soon.